Okay, great. So I'm going to go over the SV40 multiplication cycle handout. I think going through these handouts are a good way to study, simply because there's not too many words compared to lecture notes. There's a lot of pictures, but if you can explain what's going on in this whole handout, you will be able to demonstrate to yourself how much you actually know about the virus and so forth. Because when you see something like H2A, you might be like, what's that? What is that? And by going through handouts, and you do that in addition with the lecture notes, you can understand, you get a visual aid in seeing what all of the different proteins are and what all of them do, and you get to see visually what the multiplication cycle looks like. Okay, so I do not want that one red. Okay, so I'm on my computer right now, so I won't be doing much writing. I'll be doing a lot of circling. Hopefully I don't mess. I'll try to be as accurate as possible. So here goes. So right here you have the virion, obviously, and VP1. These three proteins right here, VP1, VP2, and VP3, they are the capsid proteins. VP1 is a major one, so that means that it's the one that makes up the bulk of the capsid protein for SV40. VP2 and VP3 don't make up as much of it. Now VP1, if you take a really careful look right here, you'll see VP1 right here. So that means that VP1 functions, in addition to being one of capsid proteins for SV40, it also functions as one of the antigens to interact with this MHC class 1 molecule of the host cell, so that the virion can penetrate the cell. So VP1 attaches. It helps the virus attach. Okay, so in VP2 and VP3 layer, you'll see that, or it should be written right here, but it functions, it's sloughed off in uncoding. Now, going back to this right here, you see that the virion has a double-stranded DNA. Now you might be wondering what the heck H2A, H2B, H3, H4 are. What are those things? Those are the histones of the virion. So because it's double-stranded DNA, it needs to be able to compact that DNA into a tight space of only 45 nanometers. And the way it does that is, first there's it takes a superhelical structure, and then it wraps those around nucleosomes, and the nucleosomes have the histones in them. And in essence, because its DNA is compacted in such a way, you can think of SV40 as a mini chromosome. So just like how we would look at something like poliovirus and think of it abstractly as a mRNA molecule, here you can think of SV40 as just a chromosome, and that has certain implications. There's two of them from what I can gather. First is that because it's a double-stranded DNA virus, SV40 is infectious with the genome by itself. So you isolate the SV40 genome. The genome is infectious. The second thing is, if this DNA, this viral DNA, goes into a non-permissive cell, it will literally transform the cell into a cancerous one. Okay, so that's that right there. And you can see right here, this is just the VP1, or this is the SV40 attaching to the MHC receptor that's right here, and it's entering the cell. See, so penetration. Now, something that wasn't written here in the penetration step is that it does this through caveolin receptor-mediated endocytosis. So first it attaches MHC, and then in order to actually penetrate, uses caveolin receptor-mediated endocytosis. And caveolin is just basically some proteins that's on like this like vesicle formation right here. So the virus interacts with the caveolin, and then it absorbs into it's absorbed into the host cell. So here you have partial encoding. And with the partial encoding, you lose VP2 and VP3, right? And then there are also nuclear localization signals. I think, yeah, there are nuclear localization signals on SV40 that then, because up until this point, even after it's penetrated, it's uncoded, it's still in the cytoplasm, but it wants to go to the nucleus in order to replicate its DNA and transcribe its DNA. So it needs nuclear localization signals. It's not shown here, but just keep that in mind. And then it finally comes in, those nuclear localization signals finally help the virus go into the nucleus. Once it's in the nucleus, you have a full encoding of, so you see this is the virus, this is the virus after partial encoding. After full encoding, you just have pure DNA. Okay, so now the thing is that with this DNA, what's cool about SV40 is that there are genes that are expressed early and genes that are expressed late. And the reason why it does that is because SV40 does not want to express the late genes until the conditions are right. So you notice the late genes, which is right there, are your capsid proteins, VP1, VP2, VP3. Here, your early proteins are T antigens. So what the T antigens do is that they help they allow SV40 to force the host cell to proliferate. And when a cell proliferates, it has to turn on, its cell machinery. It needs to turn on its replicative machinery, its DNA synthesis machinery. It has to start translating proteins. Obviously, SV40 is not going to let the cell make cell proteins. It's going to trick the cell into making proteins in DNA. But by tricking it into doing that, it will force the cell, that it will tell the cell to make the viral proteins in DNA. So these T antigens force the cell to start growing, start dividing, start making DNA, start making viral DNA, viral proteins, or specific viral DNA. So after the viral DNA is made, you have a bunch, tens of thousands of copies of the DNA. Now you can have a bunch of transcription translation of that DNA, and then you have VP2, you then have late gene expression. So again, the genome of SV40 is circular double-stranded DNA, and there are genes that are expressed early and genes that are expressed late. And genes that are expressed early are T antigens. The genes that are expressed late are the capsid proteins. The T antigens tell the cell to make DNA, to make RNA. And the late genes, the VP1, VP2, VP3, are only made after you have a huge amount of viral DNA and RNA. So, if you look in this next step right here, this next step, early transcription. So SV40 exploits RNA polymerase II to make, to do early transcription. So this is transcription before the SV40 has cloned its DNA, right? Because it needs a bit of the T antigens to get everything started. You can't have, without the T antigens, the SV40 cannot replicate its DNA. So in order to make the T antigens, it needs to go through early transcription. In order to go through early transcription, it exploits cellular RNA polymerase II. Right here is this illustrates that, you have made your RNA transcript of your T antigens, of your early genes right here, and it undergoes splicing. So what's cool about SV40 early genes, and really all of its genes for that matter, is that it uses alternative splicing to get different polypeptide products from the same sequence of DNA. Okay? Let me repeat that again. It takes SV40, can use the same DNA sequence to make different polypeptides through alternative splicing. Okay. Now, as a side note, that explains why its genome is so dang small, compared to something like poliovirus. Poliovirus was 7,000 bases, and yet it only encoded a few. I think it was like 5 genes, and that's all it could code for. Whereas SV40, because of alternative splicing, its ability to use alternative splicing, it's able to encode many genes in a much smaller genome size. Okay, so right here, this again shows my whole point through all this is that it does early transcription of its DNA, and then it splices that RNA transcript. Okay, now, obviously you can't make proteins in the nucleus. You need to get that RNA transcript out. So you transport it out into the cytoplasm. So this is a small antigen, large antigen. So if you notice, the small antigen, it has this thick black line right here, just there. And a large T antigen has a thick black line here, and another one right here. And this just illustrates again the fact that it's the same sequence of DNA, but because of alternative splicing, you get different polypeptide products. Okay. So you translate this. Okay, great. And this shows that some of the large T antigens goes to the plasma membrane. Now you might be asking, well, what the heck does small t and large t do? Okay, well, I will tell you now. Small t antigen binds to protein phosphokinase 2A. What the hell does that mean? Speak English. Basically, small t antigen tricks the cell into going to S phase of the cell growth cycle. Tricks it into a small t antigen, tells the cell to go into synthesis phase. Small t antigen tells the cell to start making DNA. Okay. Large T antigen does a few more things than that. Large T antigen tells the cell to assemble the DNA synthesis machinery so that it can start making viral DNA. It also unwinds the viral DNA for replication. And then it also binds to tumor suppressing genes. In this case, retinoblastin, which is a tumor suppressor, so that it can increase cell growth, right? So, if you increase cell growth, when a cell's growing, it needs to make DNA, it needs to make proteins. Large T antigen sets the stage up for the virus to take advantage of that cell growth. Okay. Large T antigen also binds to P53, which is another tumor suppressor gene. Okay. So, tumor tumor suppressor genes are there to stop the cell from growing, because you don't always want the cell to be growing. Okay, because if the cell always grows, it might become a tumor, or it just might grow and destroy the tissue type, you know. But SV40 doesn't care about what happens to us humans. It just wants to multiply. So by binding to retinoblastin and binding to p53, you can increase cell growth. And if you increase cell growth, you increase viral proliferation. P53 specifically, though, it stops the way it stops tumor cells, or preventing a cell from becoming tumor, is that it actually apoptosizes the cell. So, and it apoptosizes the cell in the context of a viral infection. So when your cell becomes infected with, polio, for example, or maybe just like the common cold, or even SV40, your cell will increase P53. That infected cell will increase P53 so that it can commit suicide, so that it can stop the virus from, it can stop the virus dead in its tracks when it's trying to multiply. However, SV40 combats this problem by making large T antigen to bind to P53 to stop apoptosis. And if you stop apoptosis, then the virus is now free to replicate a bunch of viral DNA molecules and make a bunch of viral protein molecules. Okay. And finally, large T antigen binds to P300 to stimulate expression of some genes and to stop expression of other genes. Obviously, SV40 wants to it wants to express some genes. It wants to express the right genes at the right time. It doesn't want to express everything at once. It doesn't want to express nothing all the time. It needs to express certain genes at certain times. Okay, so that explains the small T antigen and the large T antigen. Now, if you see here, the large T antigen, Dr. Samuel wrote that 95% nuclear localization. Basically, it's saying most of the large T antigen that's produced, that is produced during RNA transcription, goes back into the nucleus. It goes back into the nucleus because most of its functions are related to driving DNA replication, which you see right here. Okay. So then you make a bunch of, so you see, you replicate a bunch of double-stranded DNA, you make a bunch of double-stranded virus DNA. Now you have late transcription. In this late transcription, you still have splicing. So late transcription of the late genes. You have splicing those genes, of those RNA transcripts, bring them back outside into the cytoplasm, and then you make your capsid proteins right here. See, translation of the capsid proteins. And those things will then come back inside into the nucleus right here. And hey, guess what? Now there's a bunch of capsid proteins. There's a bunch of viral DNA. What do? Well, you put 2 and 2 together. Now you have virion assembly. Okay, a bunch of them. You just release, just like that. And that's the SV40 multiplication cycle handout. Okay, so if I had to summarize this, I would say virion enters cell into cytoplasm, goes to nucleus. From nucleus, it goes into cytoplasm, then back into nucleus, and back into cytoplasm, back into nucleus, back into cytoplasm again, and then finally outside. And remember, just keep in mind that the reason why it has to go into the nucleus and back out into the cytoplasm, back and forth like that, is because it's a DNA virus. Okay, so hope that helps. Yeah, so we're going to move on to the next handout now.